So welcome to the webinar, everyone. Today's webinar will not only showcase research from two different labs at Texas A&M, it'll also include research from the RU program in Costa Rica that Dr. Smith is a part of. For this webinar, there'll be a series of short presentations, each of which will be followed by an opportunity for questions. You can ask them live or put them in the chat, whichever is comfortable for you. We do ask that you remain muted through the webinar unless you do have a question at that time. So we are about to get started. Our first presenter is Dr. Peyton Smith. She's an assistant professor in the Soil and Crop Sciences Department at Texas A&M. And as a soil ecologist, her research focuses on how physical, biological, and chemical processes interact at soil aggregate on, excuse me, or pore scales to alter the flow of carbon and nutrients at the field or landscape scale. She also investigates how global change influences the soil microbiome and their role in the fundamental biogeochemical processes in both natural and agro ecosystems. Despite being an early career scientist, she's mentored over 30 students and postdocs, many of whom have secured successful positions in academia or governmental agencies. I'm also going to give a quick introduction about Dr. Loisel. Julie Loisel is an assistant professor of geography at Texas A&M, and she heads the Texas A&M Peat Lab, which focuses on peatlands research and climate change. If she had her way, she'd only teach field courses. She believes the connection between student and nature is essential to help them appreciate the world's intricate complexity and beauty, which leads to better stewardship. Her research revolves around carbon cycling and wetlands worldwide. And in her lab, students reconstruct past climate change using a suite of natural relics that are left in the soil layers over time, such as pollen grains, plant fossils, and many other. These climate reconstructions are then used to better understand the sensitivity of carbon cycling in wetlands over time. So I am going to let Dr. Peyton Smith get started and get your questions ready. Go ahead, Peyton.
Thank you so much, Carrie. And thank you for Carrie for putting this all together and PP Systems for hosting this. I'm very excited today to talk to you guys about some of the work going on in our lab. And we decided to shake it up a bit and have everyone give flash talks with auto advancing slides every 20 seconds. So I hope you guys are ready. And with that, I will start sharing. Here we go. Okay, so today I want to talk to you about soil that forms in tree canopies. And I'm going to be showcasing the work of Hannah Connick, who is here today. So she was an REU student in the Costa Rica REU program that's run out of Texas A&M. And it goes down to the Salta Center in Costa Rica. And so one of the things that I wanted to talk about was, what exactly are canopy soils? We're going to talk all about their carbon cycling, but what are they? They're mainly these histosols, very organic in various forms of decomposition, sometimes within suspended forms even of decomposition. So they start to build up a lot of root matter. And where they are around the world, we can actually find them in any sort of rainforest or humid forest, not just in the tropics. So you can see here, we have some along the western coast of Washington in the US, but I'm going to focus today mainly on work that happens at the Salta Center from Texas A&M. And before I start, if you're really asking yourself, how is that soil? Is that really soil? It is indeed soil. We do classify it as soil. The Soil Science Society of America does. I think the World Soil Reference Base has not yet defined it, but just recently in 2017, the definition of soils became more inclusive. And because these are organic soils and have a lot of carbon, I thought, well, how much carbon are in these soils way up in the trees? And so I really wanted to identify these source and these sink potentials and really get to this question of, like, are we overlooking a really substantial source or sink of carbon, hopefully a sink. In order to do this, you've got to climb up the tree. So here we've got these fabulous pictures. Here's wonderful Hannah right here. An undergraduate from Franklin and Marshall University or College, and Kaylor, who's climbing the tree with her, and Jessica Murray. So you can see that it's quite difficult, but you can still do transects in trees. So once we collect that soil that has been forming along those branches and trunks underneath the epiphytes and the mats, we decided to measure organic carbon, active carbon, bulk density. We looked into the molecular composition of some tree soils across Costa Rica, and Hannah even looked into some of the functional diversity in carbon cycling using some ecoplates. And of course, we want to measure how much CO2 they're giving off. And because this is about the EGM5s, you can notice we get to take it up into the canopy and take in situ measurements, but we also get to go ahead and, let's see if this will work, use it for direct injections. So our first result is that indeed there is more than 3 times more active carbon, this permanganate oxidizable carbon, in those canopy soils compared to the soils that are directly below ground. The other thing that we saw is not just the active carbon, but there's nearly 8 times more carbon in the soil of total organic carbon. And even with that higher 3 times and 8 times, you'll notice that that ratio, here's the ratio between the total or the active to total, those canopy soils are still really high. When it comes to the composition of canopy soils, we've noticed across Costa Rica, there's not too much difference. Sometimes it differs with fog, but it's quite different from those ground soils, especially when it comes to the amount of aromatics. We see a lot more aromatics in the ground soils than we do in the canopy. And the canopy has more lipids, and that could be from waxes, that could be from microbial biomass. And indeed, these soils are pretty, there are lots of different types of organisms and biomass. So even with that, we see so much CO2 coming off from those canopy soils, and yet these are all on the same order here, is that we still haven't quite yet given off more than is being stored. When we look at the functional diversity, we first saw that there wasn't much difference, but there's a lot of variation in those canopy soils. However, when you look at some of the classes, like the carboxylic and ketonic acids, we start to see some differences. And even in the carbohydrate degrading genes in the ground soil, there's usually a lot more than in the canopy soil. But again, the canopy soil has much more variation. So it's a very heterogeneous environment. The next thing is really trying to figure out these stocks, because concentrations are one thing, but to understand how it goes across the ecosystem, we need to know how much there is. And that's quite challenging with the soil in canopies relative to the forest floor. So, we do it on a per branch basis, but we need to start to scale up and figure it out on a forest basis. So take home messages are it's high in carbon, greater CO2 production, composition tied to fog. And then while that concentration is higher, probably the volume of the soil below ground is much greater. So if you have any questions, I have some resources that you can go to, some media for basic things, and some of the folks who have been working with this. And with that, I will take any questions. That goes by very fast.
So are we asking questions now or after all the talks?
You can ask questions now.
Anytime.
I have so many questions, but I'll let the audience first, and because we can catch up on campus later.
Well, I would actually recommend, Julie, asking your questions, because you may be asking a question someone else is considering. So please feel free to go ahead.
Okay, first question. How thick are those histosols, and why do you think they are preserved?
Yes, so they're pretty thick. So sometimes they can even be about 15 centimeters thick on the branch, especially on the older trees when there are a lot of epiphytes. And they're suspended because they're made up of all that litter matter, and mostly the root biomass of those epiphytes. So really, it's just kind of like collecting these, and it doesn't have quite that same biogeochemical cycles as the soils below ground. So it starts to, to just accumulate, and then it's just kind of slower to degrade. So very similar to like the peats that you probably study. These are just way up in the canopy, and they actually experience some of the variations in moisture and temperature way more than the soils do, below ground.
Yeah, and that's why I was curious, because or maybe it's the fog that keeps them moist, but in order to accumulate the organic matter, you need to have a positive water budget in those soils. And given that they're probably subjected to so much variability and they're in the tropics, I'm surprised. So yeah, it's cool.
Well, so Jessica Murray, who's at Utah State, she is a graduate student with Bonnie Waring, who's at Imperial College, and she is looking at them across an elevation gradient in Costa Rica, and she's has measured a variety of different things. So even things like, the fog, the precip, DBH, diameter of that tree, of the branch, of the angle of the branch. And really fog is one of the only variables that starts to correlate with how much soil is there and how much carbon is in that soil. So
that's cool. That makes sense. Yeah. And then it looked like the microbial diversity was less than in the actual soil. And that could also explain perhaps some of the lower decomposition rates. So yeah, balance, right?
And so what's interesting though, is I think that a lot of times those functional profiling plates that we use, they really are kind of geared more towards like a mineral soil cycling. And up in the canopy, we really have a lot more mycorrhizae and a different diversity of microorganisms, because I would argue that actually the tree soils have, and some research has shown that they have actually greater biomass and diversity for the microorganisms than the soils below ground. But I think it's just different composition of the carbon. So I think also thinking about some of the carbon in the trees, you're dealing with those fresh litters, the detritus. And I think sometimes it actually will take more energy, maybe more CO2 to decompose that than when it's below ground. And you have a lot more of these shredders and other biota that kind of puts it into smaller pieces. And even thinking on a molecular level, kind of smaller molecular pieces that are easier to turn over. That's what I think. That's what I'm thinking is happening.
It'd be cool to measure continuously what happens. So you need multiple EGM5s is what
I know. I just need to like hook them up and keep them in the trees all year.
Yeah, to know the daily variations too.
Yeah.
There's quite a bit of a difference depending on when the fog comes in and whatnot.
Yeah.
It's kind of cool. Thanks for answering all my questions. And I saw somebody was chatting, so there might be something else in there.
Okay, I see that. Oh, the carbon and nitrogen ratio compared to the ground. So there actually is, let me pull up the table. It's much higher than the ground because we've got so much more carbon compared to that nitrogen. Some canopy soils have reported high nitrogen, but the majority of the papers that I've looked into, that carbon and nitrogen ratio is so much higher in the canopy than it is below ground. The microbial composition was another question, and that for sure. So we're seeing a lot more, these more fungi. So these are epiphytes, and so we're seeing different types of mycorrhizal fungi. Sometimes there's these orchids that are epiphytes, and so orchids have their own unique mycorrhizae as well. And so I think that there's some more specialization happening up in the tree canopy than there is below ground. And the fungal to bacterial ratio is much higher than the soils, at least at the sites that we studied. All right. Do
you have any more questions, or shall we move on to the next? I do want to remind everyone that if you think of a question later, feel free to send it to me and I can forward that on.
And I am so excited to present Andreas. He is at Humboldt State University. He is an undergraduate student who will be graduating soon. And he worked with me in 2019 through the Costa Rica RU program that Dr. Moore and Brumbelow are the lead PIs of. So this is an NSF funded program. And Andreas, it was so wonderful to work with him. And some of the things that I thought that he did so well was, one, he is incredibly enthusiastic and hardworking. Unlike some of the other students that had kind of a small sample load, he was really helping out Kathy with her PhD project. So when he started the project and he came into Costa Rica, he had like 108 samples that he needed to collect all and do all the measurements for in just this like 4-week time. And he just whipped through them And so it was a joy to work with him, and I'm so excited to see where he's going to end up. And I'm so glad that it's going to be in the soil world. But I'll let him tell you all about that. And so with that, Andreas, I'll sign it off to you.
Thank you very much. Here, I'll share my screen real quick and let me know when you guys can see it. Can you guys see my screen?
You're going to want to put it in presenter mode.
Cool, good. I just want to make sure. All right, cool. Let me get this back on. Okay, cool. Hello, everybody. How you doing? My name is Andreas, and welcome to my presentation on soil carbon sequestration and man buns in science. I'd like to start out with a quote that two faculty members told me just before I presented this data in 2019. My first professor said, you should shave your face before your presentation. And my advisor said, don't say that. There's always room for beards and man buns in science. And I'll never forget that to this day. So my project focused on soil carbon sequestration in Costa Rica, but more specifically at these areas of payments for ecosystem services. So these were financial incentives offered to landowners to essentially sequester carbon below ground through afforested plantations. While we looked at sites all across north central Costa Rica, I focused on a site called the Soltis Center. And this was a carbon farm adjacent to the place where I was doing my research at. And essentially, we hypothesized that these PES plantations that were afforested would sequester carbon better than aglands, but not quite as well as their adjacent old-growth conservation forests. And so we compared carbon pools across the adjacent land uses, such as agriculture, the PES plantation, which is the carbon farm, and the conservation forest. So we measured the active carbon using potassium permanganate, permanganate oxidizable carbon. Essentially, what we did is we mixed soil with water and potassium permanganate, and then we're able to pour off a supernatant. And by getting a reflectance value, we're able to get the amount of active carbon in those soil samples, which is really cool. And so what we ended up finding was that the Soltis Center PES site had the greatest amount of soil organic carbon at both 0 to 5 and 5 to 10 centimeters of sampling depths. And when we sampled the conservation forest, we found that the conservation forest adjacent to the Soltis Center had the greatest amount of carbon readily available to microorganisms, which was really interesting. And this kind of implied that this PES site may have the potential to hold more soil carbon than a conservation forest, which I just thought was amazing considering there's so many organic inputs in a conservation forest, and the PES site was afforested. But we also wanted to make sure that we were measuring the amount of CO2 being emitted from these soils through mineralization. And so using an EGM5 gas analyzer over a 24-hour period, we measured CO2 emittance from these soil samples in these septa vials, and we're able to figure out how efficiently microbes in these soils were using this carbon and how much of it was being lost to mineralization. And so it was pretty cool. The kind of information that we found was that the highest amount of CO2 was emitted from conservation forests, and we found that the lowest amount of CO2 is being emitted from this PES site, the Soltis Center. And the PES site also emitted the lowest amount of CO2 per grams of soil organic carbon, which was really cool, implying that this PES site may be the most efficient with carbon use. So in all, we found that this PES site, the Soltis Center, the soils there have the potential to use their carbon stocks efficiently and truly sequester carbon long-term, like these PES programs that were financial incentives offered to landowners have intended them to be, which was really awesome. That there's a little bit of implications that you kind of have to go in, like, why was this afforested plantation so good at holding soil organic carbon? Because we found this O horizon in that area, which is really uncommon in tropical soils. And we found out that this PES manager actually managed the site for soil carbon accumulation by adding biochar, which is really great to sequester carbon long-term in a stable way, which I was really excited about. And where this kind of led me, this last summer, I was able to work for the NRCS on Kauai as a soil conservationist, and I dealt with soil health and Hawaiian oxasols, my favorite soil order. And we helped landowners implement biochar, cover crops, and soil amendments to help keep soil organic carbon really contained in these really highly weathered kaolinite clay soils, which is super exciting. I'm also now currently in Arcata, currently helping the NRCS soil survey team, helping to develop ecological site descriptions, as well as describing initial soil mapping in Kings Range, California in southern Humboldt. We're finding a lot of ultisols with these deep, dark umbric horizons, as well as entisols on these upland marine terraces, which are super exciting. And I've always wanted to work with the tribes, and now I'm able to work with the Yurok tribe, which is really exciting in Humboldt County, and I'm helping them determine if their man-made beaver dam analogs are negatively impacting stream channel formation through sedimentation up and downstream, which is really exciting. So I'm really excited to be able to work with them, and I'm doing my first site visit out this Sunday. So a word of advice from a man bun in science like me: take your work seriously, but don't take yourself too seriously. And I'd like to invite you guys to ask yourselves in your head, what's your man bun in science, and what helps you, and how can you use it to promote diversity, acceptance, and representation to your STEM field? I think it's important to be a new and unique scientific model and make science quirky and fun in your own way. I'd also like to give a quick shout out to Kathy and Dr. Smith for making my figures, because I lost my JMP license, not working for A&M anymore. And I really appreciate you guys for teaching me to be a quirky and unique scientist, and really allowing me to throw that flavor into my work and teaching me that that's what science is all about. So with that, I really appreciate it, and thank you guys so much.
Does anybody have any questions for Andreas?
I'll ask one. So out of all the soils that you've now been able to work with over the past couple of years, do you have a favorite?
Absolutely. I am obsessed with oxasols. It was well, first of all my clothes are dyed red from this summer. And I had to throw away a lot of stuff, but I also have my Vans are still dyed red, my shoes. And I don't know, it's just really fun working with them. I just think they're really unique. And it was really fun learning about farming with them, because since they're so weathered, you just have to work those soils. And there's so much cover crop implementation and soil amendment implementation that you have to do. And it was really cool working with these like agencies like Go Farm that were really helping farmers actually work their oxasols, which are the primary kind of soils that Hawaiians are actually farming on out there, which is really interesting. Even though there's like 10 of the 12 soil orders on Kauai, which is pretty cool. So I love oxasols so much.
Hey, Andres, that was an awesome talk. And I love carbon in soils, just like everybody here probably. And I think a lot about carbon storage as an ecosystem service or a nature-based climate solution. So I'm glad you talked about PES. You probably saw, but an article came up a couple of weeks ago, either in Science or Nature, something along those lines, talking about or questioning that as a solution, especially for above ground biomass, because of potential problems with the resilience of those stocks because of climate change and fire. So have you thought about this a little bit, and how much fire is there in Costa Rica? Maybe there isn't. I know nothing about that area, but do you worry about decadal to century scale net carbon storage in those types of ecosystems?
Climate science more Mediterranean. I know if implementing PES programs in tropical places, at least it's very wet. And I think that in terms of fire and worrying about losing biomass to that sort of thing, I don't think it's as much of an issue, at least to me. But I know I would love to see PES implemented into the United States a little bit better. But the problem is that on the West Coast, at least, we have horrible fires. I mean, it was just until probably 2 weeks ago that I mean, I'm seeing blue skies now. I hadn't seen blue skies in like 5 or 6 weeks out here. It just been so smoky. But I do worry about that if they were being implemented in the United States, because we have such a fire problem, especially on the West Coast. And even on Hawaii now, we're worrying about fires, especially on the Big Island. So that is something that I worry about. But I feel like in Costa Rica, with it being wet enough, I wouldn't really worry about it that much. But you never know with a changing climate.
Yeah, thanks.
Hey, Andreas, would you like to introduce Kathy?
Absolutely. With hours and hours of me looking at Excel sheets, not really knowing what to do and dissociating, I was really happy to always have my partner in crime, Kathy, by my side. She helped me get through a lot of my data crunching, and was always making me smile through really tough times when looking at data, as we've all been there. So with that, I'd like to introduce Kathy Quinones Gonzalez with her presentation.
Thank you. And my savior is Dr. Smith, so she's the best on that. I need to share my screen. I'm having a hard time. Okay, here it is. Do you all see my screen? By in time saying that I will be presenting unraveling soil carbon dynamics in tropical tree plantations and trying to get, okay, here it is. I want to start with a little bit of information about me. I'm a first generation PhD student from Utuado, Puerto Rico, with a background in agronomy in soils. And I worked in the field for a while until taking a career shift towards the academy, where I arrived to Texas A&M and joined the Ecology of Soil Carbon Lab, where I can further my interest in soil carbon dynamics and carbon sequestration in the tropics, especially focus on the potential for soil carbon sequestration from tree plantations under payment for ecosystem services in northeastern Costa Rica. And I want to start by acknowledging all the wonderful people and agencies that were eager to collaborate and provide the resources to identify these PES and landowners and help fulfill the country's requirements to successfully complete this research. So we made a preliminary study where we use a special, a space for time approach and evaluated soil carbon dynamics under PES plantations at adjacent sites representing pasture and the historical forest. And soil samples were taken at 2 different depths, 0 to 5 centimeter depth, and 5 to 10 centimeter depth per species, per land use, and across 3 different sites. And we also measured the quantity, mineralization, and composition of soil organic matter. And for quantity or carbon sick, we measured the total organic carbon via combustion and epoxy that Andreas talked before. And we also, for carbon source, we used the EGM 5 paired with the injection kit to measure the soil respiration. And I must give a huge shout out to Andreas for managing to run all these samples, as Dr. Smith mentioned before. I also measured the molecular composition of soil soluble carbon using Fourier transform cyclotron resonance. So what did we find? Our carbon sink results showed that the total organic carbon was significantly higher in conservation forests when compared to PES plantation and agricultural sites. But when looking at the reactive fraction, pasture soils had the highest amount of active carbon at 0 to 5 centimeter depth. On the carbon source, there was no effect on the PES, on the cumulative carbon burst. There was a trend, but it was not statistically observed. But when normalized to the amount of carbon, of organic carbon in the soil, we see that agricultural sites respite more than the forest sites, suggesting that the microorganisms in conservation forests may have higher carbon use efficiency per unit of carbon emitted to the atmosphere. Our results did not show any differences on the molecular composition of soluble carbon between PES and non-PES sites. However, when we changed to aromatic compounds, we noticed higher amounts in soils from conservation forests compared to plantations and agricultural land uses. This was also reflected in the lower active to total carbon ratio seen in conservation forests relative to agricultural land uses, which emphasizes that the conservation forests may have more persistent organic matter compared to plantations or agricultural land uses. So our take-home messages are that the stability and turnover of soil organic matter is affected by plant species rather than by PES sites or land use. Conservation forests are potentially more efficient at using the carbon they have, and chemical recalcitrance is not the main driver of carbon turnover at these sites. Obviously, more research is needed, but thank you for bearing with me and listening to my presentation. And let me stop it here. I invite you to ask me any questions before I introduce Nicole Shigley for her next presentation. Thank you. Any questions?
If you had to go back, that'd be nice. And then what would you want more measurements of?
Ooh, we did go back.
Oh, you did.
Yes, so we did go back and we identified more PES sites, and we would like to measure the amount or quantify the amount of carbon that is on the different to do soil organic matter fractionation using density fractionation and quantify the amount of carbon that is in these different size fractions and see like the turnover or persistence of these of the carbon in these sites comparing these sites.
Cool. I'm asking because, you know, as you probably know, we're asked oftentimes, especially by modelers, like, how do you know this is representative of your ecosystem? Because soils are just so heterogeneous, right? And things like that. And so every time I think, oh man, if I had more sites and I could show them that, yes, there's variation, but we can explain a lot of it, that would be nice. So I was wondering if you encountered that kind of thought.
Yeah, unfortunately, we don't have the soils here yet. Like working with all the protocols to get the soils here, but eventually I hope that next year I can get to talk about that. Thank you.
Okay, it looks like there may not be any more questions. Again, if there are any, email me and I'll share them with Kathy. It gives me an opportunity to talk to her. So Kathy, would you like to go on and introduce Nicole?
Of course, yes. I just wanted to say stay tuned, and because you will get to see our lab rockstar, which will be giving an awesome presentation about all her all the different things that she has been doing, and they're amazing. So it's an honor to just be sharing and have the opportunity to be lab mates. So go, Nicole.
All right. All right, can everybody see my screen?
Yes.
Yes, okay. So go ahead. Hello, everyone. My name is Nicole Shigley, and I'm a master's student here at Texas A&M. I'm going to take a little bit of a break from research and talk to you about mentoring soil students. And I'm going to break it down into 4 different styles of mentoring, starting with mentoring in the classroom. So in the classroom, we use this as an opportunity to enhance our lectures. We have this amazing wall of soil monoliths that show off different orders of soil that the students may not necessarily get to see in the labs. As well as under this monolith wall, we have a table full of these examples that are hands-on that we can show the students and pass around for them to feel. So the students are getting multiple ways of these lectures by hearing the lectures and getting to see and feel these examples in class. Another cool opportunity we had was using that Capshire for colorblind students. So we'll move on to mentoring outside. So the first half of the class is kind of breaking down the soil characterizing components. We talk about landforms, so we go across the floodplains and up the terraces. On the terraces, we show the Katina effect with the hill slope profile. And we are from Texas, where we have a lot of calcium carbonates. So we do a lot of practicing of acid tests to find those carbonates. And here the students are breaking those cores up to find the calcium carbonates. After learning all of the separate soil characterizing components, we split them up into groups for the later half of the class and send them off to describe these soil profiles on their own. We try to give them as large of a diversity of profiles that we can find so they can take these skills from the classroom and bring those into their internships, into their job experiences. So this profile down here has a buried horizon, this one here. This profile has some good old Texas slick and slides that you can see above. And this profile here has a CR horizon down at the bottom. This was one that we had done at a local park that had a really deep mixed top horizon that we believe was brought in or maybe mixed in when that park was made by humans, as well as some experience with some biological aspects that we briefly touch on in class, like anthills bringing up that subsoil to the top, or biocrust protecting the soil, as well as getting to experience some soil structure and biology action with the roots. And we also got to see some little critters and worms in the soil as well. So then we'll move on to mentoring with outreach. This is an opportunity to not just mentor the students in your classroom, but also kind of bring in and recruit younger students as well. So these are two opportunities that I have done personally. On the left here, we have a soil workshop that we host where we bring in 4-H and FFA students that don't have much soil experience or maybe have never been in a soil pit before and walk them through how to characterize soil. On the right over here, we have a grade school STEAM fair that I got to participate in with some other grad students. And I was in charge of the soil section where I brought examples of sand, silt, and clay for the students to get some hand texturing, as well as a little container of some calcium or some calcareous soil with acid to show them the fizzing. Students always love that. And a big hit with the students was this bread tin that we made a little soil profile out of, and let the students take straws and take little cores, and they got to pull the straw out, push the core out, and see what kind of horizons they brought up. So these are great experiences and opportunities to outreach with those younger generation. Mentoring with social media is also very important. This can reach an even broader audience. You can outreach to other mentors, to other students, even to the public, or with other companies as well. So some of these that I would like to highlight. A really good resource is fortheloveofsoil.org. She has a section for soil art and painting, and this is a little snippet from her Instagram as well, of little palettes that she's making for educational purposes. She has an entire link to soil education, where she has it broken down by different soil areas of study to bring soil into other curriculum as well. And they also just did a cross-country run that they're turning into a documentary where they stop and talk about soil profile to bring more awareness to the public. Instagram is also a great way to learn about other soil jobs like astroecology, which I recently learned about and think is super awesome. It's a great way to learn about other tools and products that you can use. There's also podcasts that you can use to break down soils to the public audience, and using Twitter to engage the public audience into your research. So with that, I would like y'all to explore creative new ways to expand your mentoring horizons. And with that, I'll take any questions.
I actually have a question for you, Nicole.
Yes.
What made you want to focus on the mentoring aspect?
So this is my third year in my master's program, starting on my third year. And so I've talked about my research a lot, and I feel like the mentoring side and education side of soil is not as well, or is not as much talked about, but I feel like it's very important, especially with the new generations coming in with a lot of technology. I feel like it's really important for us to explore new and creative ways to kind of interact with those newer generations.
Thank you. And then you mentioned something, and I couldn't hear what you said about colorblind students, something that
Yes.
you provide.
I went a little bit slower than anticipated on that side. So we had this instrument here. My first semester teaching as a grad student, we had a few colorblind students that had a little bit of issues with coloring. So something that we did at the end of the semester with extra funds was buy this instrument, which is the Capture Color Munsell Color Reader. And so it's this electronic reader that you can hold up to. It's more precise with itself if you use damp soil. which is usually what you color with the color book anyways. And you hold it up and you click the little button on the side and it captures it and it'll bring up what that month cell color is. Another really cool feature of this equipment is for soils like this that have multiple colors, you can hold the camera button halfway down and it'll show you what's on that screen and you can get both colors in the screen at the same time and it'll bring you up both of those colors.
Well, that's really cool. Thank you for answering that. Does anyone else have any questions?
I actually wanted to add to that and also talk a little bit about Nicole as well. But so what was really neat is even with that automated one, I mean, all of us we had 4 different samples and everyone in the class kind of colored it with their eye and then using the color reader. And we had a, big up on the board, like all of the different ones that we all guessed what it was. And even without having any, different vision abilities, like we all come up with different color, we all see color very differently. And so it was a really fun activity in the class just to see how we all, got these beautiful soils colored, if you can see that lovely color wheel. So what I wanted to say about Nicole, she mentioned being on her 3rd year, but that is because she was in charge of even teaching an undergraduate course in soil morphology in spring of 2020. So halfway through the semester, right, like many of us, she had to completely take her class online. And it's a field course. And so she had to really come up with all these really great ways to still give them an experience, but physically distance. And she managed to do that with flying colors and has created a lot of activities. And so, she's submitting that to the online repository. So, it's not that she's, behind. She has been doing so much, not just, stellar research. And I wanted to tie that with Kathy a little bit because, well, one, you'll notice that everyone who's talked, we're all really enthusiastic about soil. Like, we're all big soil dorks, which I think is why we all decided to come work together, because we appreciate Andreas as well. We're really into lots of soil puns, but Nicole's work is on how tillage, her research is on how tillage influences soil carbon dynamics. And when I first met her and she came to my office, she was really interested in doing something like that, looking at different agricultural management practices and soil health. And at the time I didn't quite have the field sites. And I was like, I have this project in Costa Rica and I'm really interested in payment for ecosystem services and how much soil carbon. Would you be interested in that? I don't know if that's something you're interested in. And she was just very cool and calm and collected and was like, yes, oh yes, that's interesting. Okay, and I was like, oh, maybe she doesn't like it. But like, sure enough,
Just dying.
she was. So she took that project over, even though her background wasn't all these different fractions of organic matter. And anyway, so it's really fun because then Nicole comes around and now she's doing exactly what Kathy wants to do as well. But now Kathy gets to go to Costa Rica all the time, and hopefully we can bring Nicole
project
one day soon so she can characterize all of our soil there. All right, I'm done being a mom.
No, I think that was great input. Thank you. But that's what I was thinking is like she's explaining when, we were just excited she's here. But I would, if no one else has any, actually there are a couple of questions here. Someone's commenting, great teaching ideas, a fun one for outreach. They did with their students was to make pH meters with red cabbages and get students to compare their garden soils with different household items. Students loved it and happy to share resources. She's happy to share new resources. Sumant asked which soils, for example, oxysols and vertisols, are susceptible or vulnerable for climate change. I'm not sure who wants to answer that question. It is answered, all of them. And definitely gelisols. Okay. Well, if there aren't any more questions so far, Nicole, would you like to introduce Ayush?
Yes. Oh, I just wanted to, Dr. Smith was saying that I'm gonna have the resources ready. I just wanted to say they'll be submitted here on the Explore Soil Education if anybody's interested in them. So with that, I would like to introduce Dr. Ayush He is a very calm and patient R code wizard, and he has helped us all tremendously in R code. He says no, but it's the truth. And with that, I will let him take over.
Thank you, Nicole. Good morning to all. And thank you for your work with teaching and all, Nicole. I love the resources you shared as well. That's awesome. So with that, let me get started. Can you all see my screen?
Yeah.
Hey everybody, my name is Ayush, and today I'm going to try to address the sample number dilemma that we have. This is my plot when I was doing PhD in Virginia. These are huge plots, and we just took 2 samples, and I couldn't sleep at night thinking, well, is it enough samples, right? Is the power too low? So even existing studies shows that for soil carbon, the power is around 20 to 60%. What we're aiming for is about 80 to 90%. That's the probability of us making the right decision. So this is an important thing before we make bigger conclusions. So with that in mind, the objective of this study is to quantify the minimum sample we may need to quantify these soil enzymes and soil carbon properly. We have also tried to evaluate and propose a simulation-based approach for this, and also try to see how the row position sampling affects on these questions. For this, we installed 2 sites in Texas, where we installed 6 treatment, tillage as the whole plot and cover crop as the subplot factor. These 2 sites had different soil types. In each of the sites, we have 18 plots, and one plot. So this is a figure of one plot where we took samples. We took 20 samples, 10 in-row samples, which are in green, and 10 between row samples, which are in orange. So those were randomly selected sampling points even before we visited the field. So we make grids in the lab and visit field to remove bias. In all of the samples, we measured these parameters, which are soil enzyme activities, active carbon, soil organic carbon, and pH. So we try to capture parameters which have different spatial variability. And stats were done and checked for significance for these parameters. So our simulation approach is basically done in R, which mimics sampling for 1 to 9 samples per plot, as the GIF shows here. Each time it's taking 1 to 9 samples, doing stats, recording p-value, and this process was repeated 10,000 times to cover all combination. And the p-value recorded was then mapped into a violin plot, as you can see here, with sample number on x-axis and cover crop p-value on y-axis. This is an example violin plot. We had 28 or 29 plots. So it shows that 3 samples were enough to detect this cover crop effect. So using these kind of data, we then plot a bar plot, right, which has parameters on x-axis, and our simulation suggested sample number on y-axis. It shows that enzyme activities required more number of samples to detect treatment effect, which is obvious because they are more spatially variable compared to other parameters. We also tried to compare our simulation approach on y-axis to power analysis result on x-axis. This is for site 1. It shows positive correlation, and it did not affect so much between in and between row, but we can clearly see the thing that power analysis suggests more samples needed compared to our simulation approach, which is true in site 1, and which is also true for site 2, which is also a similar scatter plot, with in row being green samples and between row being the yellow samples, again showing power analysis suggesting higher number of samples may be needed compared to our simulation results. So these kind of approaches are really necessary, and it has been shown in previous research that soil health metrics and even extracellular enzyme activities in particular are so much variable, we may not be quantifying them properly before we move on to make bigger conclusions and effect management. So when we compare our simulation approach to traditional power analysis, we have some advantages, right? The cost might be more power analysis because there's more samples is needed. We can incorporate pairwise comparison with our simulation approach, and we can use our exact experimental design to be sure that we're getting exact values. And why we're trying to get this representative sample is that we need that peace of mind that we're capturing the data properly, that we all crave for that representative sample. Even random sampling may help, but it's not the answer, as you can see with a cartoon figure there. So a main takeaway is that if you want to achieve that peace of mind, if you want to have that representative sample, try this simulation approach. With that, I can take questions.
Does anyone have any questions for Ayush?
Did I do a great job?
So I'll say that Ayush had an undergraduate that he mentored through this process and who is not here today because he's busy fixing some of our equipment. But Jacob worked with Ayush and he was in charge of kind of running all the enzyme assays on these samples, in which there were more than 700 samples. So, so Jacob was quite busy during his senior year running all these assays. And hopefully you'll see this work out soon from Ayush. And he also was successful in getting a grant to study soil health metrics in different grazing management strategies as well. So looking forward to see where he takes that.
We'll have to bring him back and see what he learns.
Sounds good.
Well, I know that Ayush doesn't know Gillian personally, so we're actually going to have Julie introduce Gillian.
Right, and it's my pleasure to introduce Gillian Sanders, our next speaker. Gillian started taking graduate student seminars as an undergrad, and yet she was that student in the classroom that would look at a graph, take it apart, and criticize some very senior scientists in very high impact journals. So I've always been very excited to be working with Gillian and impressed by her capacity to really think critically. And on top of that, you know, she's a talented writer, she's smart. In the lab, she's a total tinkerer. So she loves, you know, adding a bunch of tubes and making everything work the way she wants it to work. And this is about her. But so last year, because of COVID, it was really difficult to teach her how to do these analyses in the soil. And on top of that, I had a baby, so I was not there. And a lot of what had to happen in the lab happened thanks to her. And she even mentored 2 undergrads to help her do all of the analysis that were needed. We also gained a lot of perspective with working with Dr. Smith. So she's not a R savant yet, but she is one in the making. And so I'm very proud of that. And lastly, she is a lab t-shirt designer. And I think everybody needs that. And we didn't talk, I promise, but we're both super proud of the t-shirts she made. So here it is. And without any further ado, I will shut up and let her talk about her research.
Awesome. Thank you so much. That was a great introduction. I might get my slides ready. Okay. Cool. Everyone can see this? Awesome. Cool. So I am studying Arctic permafrost carbon, and in particular, my research study is investigating CO2 production from mineral and organic permafrost soils from a continuous permafrost zone in Alaska. And I just wanted to show you all a little picture of a peat core from Alaska, so you can see all the sphagnum moss on top. Really cool stuff. Just to give some background, the Arctic is a very significant carbon sink. It stores approximately half of the global soil carbon pool in all of the permafrost zonations, so continuous through discontinuous. And also peatlands are very important climate modifiers, so they store about 1/3 of the global soil carbon pool, but they might expand or drain. We just don't really know with climate change. So this study is going to try and investigate how these peat and permafrost mineral areas might respond to climate change. I did 2 temperature treatments, and I have 2 soil types, upland and lowland, and I used my little buddy, the EGM. for the past year. So Toolik Lake is in the North Slope borough of Alaska. It's on the north side of the Brooks Range. It's very cold. The average annual air temperature is -8 degrees. And there are lots of acidic gelisols, and there's some peat muck. And actually, there's been some recent discovery of peat patches there. So that's kind of what we're looking at, both the peat and mineral soils there. So I did a whole core incubation. There were 24 total cores, kept them in 2 different freezers at 1 degree Celsius and 6.5 degrees Celsius. And we had open caps, and we did direct injection with the EGM at field moisture conditions, or as close to field moisture as we could get. And here's some pictures. I have a picture of a peat core on the left, and that was right before sampling. So it's all taped up tight. So we only capped them for an hour before sampling. And then in the middle, there's a picture of the partitioned layers that we prepared for a separate incubation, and then a picture of the gas cap on the right that I made with lots of tools. Very fun stuff. So for the partitioned layer incubation, we had 33 16 milliliter vials of broken up soil layers from those whole cores that we dried and then re-wetted and incubated in the same conditions for 100 days. Yeah, super fun. And here I just wanted to show a picture of our sampling setup. So we did the direct injection kit on the EGM, but we attached some tape and a lure lock to make it work better for us. Because as you can see, our gas caps also had a lure lock, so we needed to use a syringe without a needle. And Peyton helped develop this, so she's kind of the pioneer of the lure lock direct injection. And I wanted to show a picture of a typical peak from a peat core after sampling, after just being capped for 1 hour. This particular peat core had almost 1,600 parts per million of CO2, and that is pretty standard across the experiment for some of these peat cores. So lots of CO2 hasn't been analyzed yet in the lab with statistics, but that's what I'm working on right now. So I finished up soil deconstruction, and now the samples are being analyzed in Dr. Peyton's lab with an elementar. And slowly but surely, I'm making my way through the data analysis. So we should have our results at the end of the semester. I'm very excited to see what the difference is between the peat and mineral cores. So overall, we're hoping to help improve soil decomposition models in the Arctic, looking forward toward how peatlands are going to respond to climate change. And this will also help predict future carbon budgets, and hopefully that can help provide information for climate mitigation efforts. So yeah, any questions? Cool.
I do think it's cool that we cover everything from the tropics to the Arctic in this. I think that's pretty interesting. But if anyone has any questions for Gillian, grab her now.
I think we should run some of that FTICR on your peat soils, and we can look at how organic soils are different from, permafrost affected versus the organic soils we get in the canopy.
That'd be super cool. I know Julie was kind of already introduced by Carrie, but I just wanted to help segue her a little bit since she gave me such a good introduction. So Julie is basically the peatlands queen. She seemingly knows everything about peat. Literally anytime I have a question and I can't seem to find the answer online, she somehow knows the answer. So she's a fabulous mentor. She knows her stuff. And even though, she had a baby last year, she was always there for me when I had a question. And I would not have been able to be successful if she weren't my advisor. So without further ado, go Julie.
Oh, thanks. Let me share my screen here. Okay, and put this into a better format. This thing is in the way. Okay, here we go. Thank you so much for the introduction, giving me the chance to talk here. If I had it my way, I would always be in the field. I love taking students in the field in particular because they can really take part in the scientific process. And in case I run out of time today somehow, the bottom picture here is what it is all about, getting the students' hands dirty. I'm new to measuring soil CO2 efflux, and I just purchased an EGM5. So right now I'm thinking about what is it that I can do outside and with my students that would utilize this new instrument. So today I'm going to share 4 tips, or really general guidelines to try and guide those who are trying to keep their students engaged. And I hope Nicole will approve. So wherever you are, in beautiful estuaries, by the water, in gardens, if you're in an urban setting, or if you're lucky enough to be nearby mountains, I think those rule of thumbs will apply to all of those cases. And to me, the biggest fear is student boredom. So there's one instrument and there's 30 students and everybody's just like staring at a hole and doing nothing or taking a nap, God forbid. And so the EGM5 gives me a bit of that anxiety. So I spend a lot of time thinking about what else could the other students do so that we have a more active and engaging experience. Oh, and here's Gillian. So the first tip, or really general guideline, is to keep the students busy, not just with measuring the soil, but at the same time, they should be gathering extra information about the ecosystems. In this case, she's coring a tree. You'll see a couple more students in a second that are doing the same. And I love the coring tree aspect because it gives you an idea of the age of the site, and then you can start comparing multiple sites and the relationship between the age of the stand and the soil respiration, for example. But it could be vegetation releves, it can be digging soil pits. That's another great example of keeping everybody busy and engaged. My second tip has to do with maintaining or keeping the students interested and engaged throughout the whole process, not just the actual measurement, because this way they really get to appreciate the true value of having a kind of an outlook on the scientific process. So I ask them to always develop their own hypotheses before we reach the field. And then we need to adapt them sometimes in the field. And after that, I have them draw some of their hypotheses and analyze some of the results as well, and mostly anticipated results so that they really have to think through the whole process and what they think they will get out of the experience. Oh, I'm talking so fast. Yes, the third one is that there's always troubleshooting in the field. And so I usually ask them to solve the issues. In here, Gillian again on the right, these students were trying to take a core in that wetland and we couldn't push it, the 5 of us. So we decided to take Lauren and stick it on top of the coring device and then she could jump. So this way we managed to get it deeper. In that second case, we are in the high Arctic in Canada. And the weather station was getting its wires eaten up by foxes. And so the students created a label that says, keep the foxes away or something. I can't read it from here. And it worked. So we have 2 years of data and the tape kept them away. And my last tip, or is I think the most important one, and the reason why I love the field so much, is that it gives you an appreciation for how complex the world is, how little you are in the big picture. And just this general awe of nature that is so important to experience and that is really hard to transcend in a classroom setting. And FYI, all the pictures I showed are from different sites that I've had the chance to visit with students throughout the past few years. And I think that's what gets to them too. And they come back transformed from any field experience because they really get to see the beauty of it all. So as a conclusion, just get everybody's hands dirty and perhaps even their faces if they want to. So thank you.
Does anyone have any questions for Julie? That was a great presentation, by the way.
Thank you.
I have a slightly unrelated question, but I'm curious if the Dr. Peyton lab can get some of those Pete shirts. I love those.
Thank you. Well, you have to ask Gillian. We do have a few extra.
Yeah, we have a few extra, but we could also put in another order and like put an order form link on Instagram if there's enough interest. Y'all should see the back. Hang on really fast. There's like a peatland model. I don't know if you can see it super well, but a student actually made this graphic and we put it on the t-shirt.
Yeah, it's an infographic that shows the agents of change impacting the carbon sink capacity of peatlands around the world. It's really cool.
We need to make a lab shirt too. We'll exchange. We'll do a shirt exchange.
I love it.
Sounds like a side hustle, Gillian. I'll help you. Well, if there aren't any more questions, I reached out to the students. As I said initially, the research is important, and the reason the research has been so good and you all have thrived so well, because not all labs did, was it really had to do with what was at the core of this, which was the mentoring and support. And I actually privately reached out to the students because I knew you wouldn't let me if I told you. So I did want to say that there were some personal comments about each of you individually, but there was a reoccurring theme throughout, and it was that you were brutally honest and very encouraging. So there's an incredible respect for your students, that you're not there to sugarcoat things and really help them. That no matter how busy you are, you take the time to meet regularly and follow up, and you're very transparent in your conversations, which is very much appreciated by the students. And they all said how you're constantly looking for a teaching moment, and you're constantly finding ways to grow and push, encourage, and highlight what they're doing, and teaching them about showcasing themselves as a team and individually. And it's sounds like every lab would have that, but they don't. So for the professors that are listening, listen up. And for students that feel like there's something missing, or they're looking to join another lab, look for these features in your professors. I can say that because nobody can come after me for that, but it's true. But I did want to make sure that that was mentioned, because it's meant a great deal to the students, and it comes organically for you. So I just wanted to make sure we shared that, because I did think the mentoring was one of the reasons that it attracted me to the group. I've known Peyton for a little while, and I'm just getting to meet you, Julie, but you guys share a lot of the same qualities. So I just wanted to make sure I shared that before we finished up.
Thank you. We all have such wonderful students too.
Yep.
Such hard workers.
Absolutely. Yeah. Yeah. It's all fun and everything, but they do hard. We work very hard. We push them hard.
But it's appreciated. The process and the respect is appreciated, because you're trying to make them better at what they do. So we just wanted to make sure I shared that, because I think that's, in this day and age, it was a very difficult year, and not everyone did well. And your groups really did. So I'm sure they're also super bright, but there definitely was some really important mentoring. So I want to make sure I shared that. If no one else has any questions, I do want to tell you that if you have anything that you think of afterwards, you can email me. We will send a recording to everyone. And I sent an email to each of you, if you could take a quick look at it once we finish here, because I'd like to recap with you and get your take. If anyone watching this has any input for me, I'm open to it. I'd like to do more webinars like this. I think it's important to show the research and the entire lab, not just everybody at the top, because I think everyone's important. So thank you to everyone for your time. We will make sure to get links out to everybody, and I had a great time. I hope you all did. So have a good day, everyone.
Thank you for attending this webinar. To learn more about our soil solutions, visit pbsystems.com.